This is the Convergent Science Network podcast. Leading researchers in the domain of neuroscience, brain theory, and technology are interviewed by Paul Verschure and Tony Prescott.
This is Paul Verschure and Tony Prescott with the Convergent Science Network podcast. And in this episode that we record as part of the Convergent Science Network Barcelona Cognition Brain Technology Summer School, we're talking with Nick Strausfeld. So Nick, you started your talk with this notion of placing the study of biology always in the context of evolution, because it wouldn't make any sense. So where's that coming from?
Well, it's coming from Theodosius Dobzhansky's statement, which he made in the 50s, and really very, very, I think was very relevant to what was happening in biology then, which was a lot of comparative work being done in all sorts of different fields. And it's worthwhile reminding people today of this because we become so incredibly specialized and canalized in our research. So there's these horrible things called model organisms, which I loathe the term model. These are animals. But there is about 3 animals that have been funded, at least in America. And you can't really tell anything about evolution from the 3. And evolution will tell us about very, very interesting aspects of design. And I don't mean that in the, in the bad sense. I mean the results of an evolutionary innovations, and what was the beginning in the nervous system, for example. What were the basic circuits that provided an organism with choice, with allocentric memory, with the ability to integrate odorant signals to provide behavior, and basically all the kind of senses it has, and what kind of nervous system allowed the integration of those senses to provide elaborate behaviors, and one is only going to get to those answers if one actually does comparative work and thinks about how these things have evolved over time.
But then, so where do you stand on that right now? So you're one of the world's experts on definitely the insect brain. So how should we think about the insect brain? What are then these basic design features it has? What are the basic functional capabilities that are supported with it? How should we think about that?
Well, we have to start thinking about where do insects come from? And the evidence is very strong. They have originated from marine ancestors, which we call the crustaceans. And then one has to ask, what is common between insects and crustaceans? What did their common ancestor perhaps look like? And if one can then, resolve the commonalities of the two, then one has an idea of the ancestral brain that common with the insects and crustaceans. Then one has to go even deeper in time and ask, well, what about the ancestors of that ancestor? When did the first insect-like brain or the crustacean-like brain evolve in geological time? And that's much more difficult, of course.
Right. But now, what are the core structures in that brain that you think we should be focusing on in that exploration?
Well, you can ask questions about the core structures, say, of a particular modality or sensory organ, such as the compound eye. So what are the core structures of the compound eye, the core circuits that allow motion detection, for example, motion vision, figure ground discrimination? You can actually resolve some of the neurons that are involved in those computations in a fly's eye. And then you need to get to the actual circuits that provide those computations, not just neurons that encode those computations. And that's, again, more difficult. So the search image could develop from looking at much simpler systems. What do they have in common with the fly system, for example? And so if you compare across many, many different taxa, say, of flies that have, they have ballistic flight, flies that can hover, flies that do this, that do that. And then you ask, what do they have in common, all these visual systems? What are the common denominators, if you like? And if you find those, then you can ask questions about, are those really the principal elements that provide information about motion detection? And then go to the crustaceans, do those exist in crustaceans? And then go even to more simple organisms such as centipedes and so forth.
But then if you talk about search image, you really mean a structural template. It's an anatomical template, or is more than that?
The search image develops from really understanding at least one circuit in one organism, and you ask then, is that circuit or are those neurons visible, or are they obtainable in other species? That's the search image.
Mm-hmm.
Yeah.
But now, where do you see the origins of that nervous system? Because in some sense, you could also argue, look, it all started with, let's say, chemical sensing, and the rest evolves from that point, right? So
yes. I mean, I would envisage that the chemical senses are the earliest. Before there was highly developed vision, but the actual computational circuits that provided choice, behavioral choice, or for example, memory of place, they can operate equally well in a chemical environment as in a visual environment or even a tactile environment.
Right.
So the basic organization of the brain was, one could say, it was independent of the modality, because it wasn't, because certainly it had to evolve in conjunction with the present modality.
Right.
But it can employ other modalities. In fact, we have a very good example in one species of insect where the mushroom bodies, which are in Drosophila and many other insects, they get their one of their major inputs from the olfactory system. In a species of insect called whirligig beetles, they completely switch modalities and get all their inputs from the visual system. So if these are place memory centers, which we think they are, then in one instance they are using olfaction and the others are using vision. You just get this switch.
But then that would imply that you're thinking of some sort of backbone structure,
yes,
prototypical backbone structure that you can sort of mold to, let's say, the sensory ecology, the behavioral ecology of the organism. So what is that backbone structure?
So I'll give you an example. The olfactory system of insects and also of crustaceans, the first synaptic neuropil is a system of glomeruli. And in insects, each glomerulus receives the inputs from a set of olfactory receptors that encode the same kinds of information of the same kind of ligands. So one type of olfactory sensory receptor neuron will send its axon to one particular kind of glomerulus. So when you have 40 different kinds of receptors, you have 40 glomeruli, each getting inputs from that particular type of receptor, right? So when you go to the visual system, you see the remarkable thing that in the next ganglia up, which is the protocerebrum of the insect, you have a glomerular organization into which are segregating pathways in the visual system. And this suggests that you have this template of computational entities, which are repeated segment for segment, which can deal with the visual input or with the olfactory input or with the tactile input. But the actual computational network is in principle the same for any sensory modality, which is remarkable, and this is quite worrying in a sense.
Right. So, but in this example, you would say, I guess the lobula would have a glomerular.
No, definitely not.
Okay.
The lobula is composed of many palisades of neurons, types of neurons, and each palisade encodes a visual primitive, such as an oriented edge of one kind or the other. And each of these palisades then sends its axons convergently to a glomerulus. So each glomerulus represents a particular visual modality, an edge or a color or whatever.
Right.
Then interactions amongst these glomeruli further reconstruct the visual image and further provide higher order primitives. The same is happening in the olfactory system. So each glomerulus receives information about a small sort of palette of ligands, and interactions amongst these various glomeruli provide information about the odor.
Right.
Not the odorant, but the odor, the combinations. So it's the very same principle, actually.
No, I understood the principle. I'm just trying to place it in this visual hierarchy, because in the chemical sensing system, it's just one step away from the receptor.
Exactly. That's the question that is always addressed. The homologue, if you like, the functional homologue of the antenna in the visual system is the entire retinotopic mosaic down to the output of the lobula.
Oh, you mean the antenna in the chemical sensing system?
Yes, it's the antenna in the chemical sensors. The equivalent of that in the visual system is the lamina medulla and the lobula.
Right. Okay.
And the outputs from the lobula are equivalent to the outputs from the antenna.
All right. Okay. So then, where would I find the glomerular structures of the optic system in the insect?
Beneath the lobula.
Okay. Very good. And you will find also an equivalent to something like a projection neuron type readout of these.
Yes.
These would be your white field neurons, or
yes, that's what we're looking at right now.
Okay. Yeah. Very good. Yeah. That's beautiful. But then, in some sense, we're still talking about the sensory front end of this, right? So the core structure, this archetypical backbone structure, should also be relying on structures that are mapping now sensor states into action. So
you're quite right. So you have these reiterated ganglia down the rest of the body, And in each ganglion, you have domains, glomerular-like domains, and each domain is receiving one or another kind of sensory input, often mapped. If it's an input of the mechanosensory system, then that particular type of receptor will invade one of these domains, and you'll have a map representation of those terminals in the domain. And then you have local interneurons linking these various domains and integrating all this sensory information, just as it does in the olfactory system or the visual system.
But what happened to these really very central structures like mushroom bodies or protocerebrum? I mean, they're sort of in between that mapping, right?
And they're unique to the first segment of the brain. And where they come from is a real puzzle. And my suspicion is that they are actually derived from the ancestor of the lophotrochozoans and ecdysozoans. And you see mushroom bodies and central body complexes in worms, in polychaete worms. In the what's called the acron, which is an asegmental neuropil. It does not belong to a segment. And so the idea is, and there are many opponents to this idea, the idea is that the modern arthropod brain is actually composed of 4 components. 3 of them derive from ganglia, primitively from ganglia, and the 4th component right at the front is the leftover from the common ancestor. And you still find this in polychaete worms separated from the rest of the system. And that's been integrated into the first segment of the insect or crustacean brain. And it is the substrate for the central body complex and the mushroom bodies.
Right. And that then provides you, let's say, the integrative infrastructure for these different sensory modalities.
It provides the substrates for high-level computations of allocentricity, a relative memory amongst individuals, and also behavioral choice, and things, complex things like dexterity and such like.
Right.
Yeah. This is the brain within the brain, if you like.
Right. Okay.
The integration amongst sensory systems can be quite local to provide reflex actions, but the integration amongst sensory systems to provide these more complex behaviors requires these higher centers.
Right. So now we have a bit of an idea what sort of the key components of such an archetypical brain could look like. But you also emphasize that, let's say, the specifics of the ecology of the animal might lead to variations now in that design.
Yeah.
So how does that really happen? Is it only through, let's say, evolutionary pressures, or over long time scales, or are there more rapid ways to, let's say, reconfigure and configure these prototype brains?
We don't know how plastic they are,
right,
in terms of the lifetime of a single organism, but with regard to the structures and the number of neurons and the connectivities, the differences between taxa are clearly the result of many millions of years of evolution, divergence.
But still, you will find also slight deviations within specific taxa or species.
Oh, yeah.
Dependent on the ecologies they're in,
well,
right?
Yeah, the difference is that's the stuff of evolution. I mean, you look at the olfactory lobes across lots of Drosophila and you find all these sort of variations of wiring. But you've got to have this. Otherwise you would not have selectivity. You would not have, the species responding to selective pressures. Some of those variations are fitter, greater fitness than others on the organism. Yeah, you must have variation.
No, but how do you see that exploratory mechanism? Because I mean, within every species, there's in this backbone neural structure that gives it the nervous system, there are exploratory mechanisms that now are imposing variability on this. And for instance, in your talk, you mentioned that possibly a computational substrate for this kind of variability in, let's say, visual processing could be the lobular plate. Because there you can start to play with how you integrate this kind of information. So would you then predict that also at the level of the lobular plate, you would find the highest variability to allow a species to explore, let's say, the variability of its visual space in which it has to behave?
Yeah, that's true. And in fact, when we look across different species of flies, for example, which have actually very different visually induced flying behaviors, we do find very, very fascinating differences of the organization of the lobular plate and the lobula, but not the medulla and lamina.
Right, okay.
Yeah.
What about the role of development here? Because I've heard, for instance, in the locust, a significant effect on the development of the brain, for instance, due to whether or not the animal is going to be part of a swarm because there's a high density of population, or is going to be perhaps more of a low density individual locust, and being part of a swarm somehow makes the locust develop a larger brain.
Yeah, that's a very interesting point. Actually, it's fascinating. What's happening in the locust? Are the neurons actually sprouting more dendrites? There's certainly a radical difference of size of the brain overall. Does that pertain to all the centers? Does that pertain to certain neuropils? I would put my money on the central body complex and its associated structures, because that's the decision-making part of the brain. But I don't think it's been followed through yet, and I look forward to seeing papers that are following through on this. But it's very, very, very interesting.
But it does suggest that the real potential for the environment to affect the developmental process, so that some of these differences we get are partly due to maybe some genetic change, but also when the environment changes, that there's plasticity in the development of the system, which can really result in some important changes.
One puzzle, I think, is that these changes happen before the locust becomes either goes into the swarming mode, or the actual wandering, continues the wandering mode that it had as a juvenile. So it's as if the brain is preparing itself for the next role, which is really amazing, and it must be under some kind of hormonal control and so forth. But again, I think we know too little about what is actually changing to put one's finger on why it's changing. It is a pity. I'd like to know that.
So what struck me about your talk is that one of the things that really fascinates you is to trace back modern brains or modern nervous systems to what the ancestral brains might have looked like. And of course, that's a task where you have to look at indirect evidence, because there are lots of clues, but nothing definitive really that can tell us what was the path that evolution followed. So can you just summarize quickly the range of clues that you think are important to reconstructing evolutionary history here?
Yeah, well, the clues must be constrained to what one would imagine would be visible in a fossil. So isolated neuropils, one from the other, that are connected by tracts, for example. One would be able to see these clearly in a fossil if they existed. Internal structures of the brain would be very unlikely to see them, although the brain I talked about today, which is 535 million years old, in a stem group arthropod, we can actually see differences of texture within the outline of the brain. That suggests, for example, the olfactory lobes, and it would be nice to see then other striations or pronounced, maybe outlines, internal outlines that would suggest the presence of certain centers that we know are required for certain functions of modern of the behavior of modern taxa. Another thing we would look for is the kind of fusion of segments that comprise the brain. In modern crustaceans, melanocultural crustacean insects, there are 3 segments that are fused to comprise the brain, and in some species, even the subesophageal ganglia, the 3 subesophageal ganglia, have moved up and have become almost fused with the supraesophageal ganglia, so that the gut just penetrates the brain. There's no clear divisions of ganglia. The whole thing becomes highly condensed, which is a mark of modernity, perhaps evolutionary modernity. Now, I would like to go and look at these ancient brains and ask: is that true? How much condensation of the nervous system can one see in the head? And for the animal I was showing you today, it's surprising that the first 3 brain fragments are fused. We were really surprised to see this. Quite shocked, actually. But that shows us it's already quite advanced.
So you're talking about the sort of anatomical, morphological markers.
Yes.
Now, nowadays there are a lot of molecular tools looking at DNA, RNA, and so on that can tell us a lot about the relationships between different animal groups.
Yes.
But I think you were persuading me in your talk this morning that this isn't enough. Although we might get some idea about what's related to what, if we really want to understand the history,
yes,
we do need the anatomy.
Yes. A case in point is the origin of insects. Did they derive from a branchiopod-like ancestor, or a remipede-like ancestor, or a malacostracan-like ancestor? The molecular studies suggest malacostracan, remipede, but there are other molecular studies which would equally well suggest branchiopod. One wouldn't know unless one went to the morphology, and morphology suggests brachiopods are very simple, therefore they must be maybe more primitive than insects, which are more complex. Which then suggests that insect brains and malacostracan brains have evolved by convergence, which is probably nonsense. So one needs these various strategies for looking using cladistics to see whether or not there's evidence for reversal and for loss of structures. That gives the false impression of primitivity. And one cannot possibly do that just using molecular techniques. One has to go in there using anatomical methods.
So there is this bias I think we have to think that things get more complicated over time.
Yes.
But in fact, there's evidence that in some cases things will actually get simpler.
Like tapeworms, for example.
So can you give an example of a reversal?
Well, parasites. I mean, tapeworms are levotrochozoans. They don't even have a brain. I mean, they've lost everything, really. And they're just reproductive organs, which are living off one's gut, I mean, there are lots of nematodes. There's another. These are ecdysozoans. They molt. Presumably their ancestors had worked somewhat more complicated.
So that means there
are a lot of examples of reduction and loss.
But also, as Tony mentioned earlier, this sort of like simple heuristic that would say, look, brains go from simple to complex is not working. It's not as a guideline. It's not helping us.
No.
But what should replace this heuristic? What's the heuristic that we should then follow?
That's a difficult question to answer.
No heuristic.
I don't know.
I think Stephen Gould compared evolution to a drunk man stumbling along the pavement. So sometimes you move further out from the wall, which could be an increasing complexity. Sometimes you move closer to it.
That's a nice analogy.
Yeah. So, and another interesting thing I think that came from your talk, which perhaps also links with some of Stephen Jay Gould's ideas, is that we, I think, used to imagine that evolution was a slow process and a gradual process. And over hundreds of millions of years, we got these advanced forms or these recent forms we see today. But actually, what was striking about your talk today was that you were arguing that some of the things that we see in contemporary organisms were already present really shortly after the appearance of the first multicelled animals.
Yes, shortly meaning yes.
Well,
yes. 50 million years is a very short time. Yes, exactly. So this is really quite fascinating. You have this plethora of different body forms in Cambrian, called the Cambrian explosion. These different arthropod-like animals.
And this is 500 million years ago.
Suggesting very, very rapid evolution of morphologies. And yet, the ground pattern of the nervous system, probably across them all, is this very, very consistent organization that exists today. So you have, in a sense, stasis and slow elaboration of the ground pattern of the nervous system, and then this very, very rapid elaboration of appendages and other decorations that provide the inputs to this consistent organization of the CNS.
Would you argue that then still that very basic proto-brain was the facilitator of all this variability?
No. It had to accommodate this variability through evolutionary time. I don't think it was the facilitator. Although one can't really imagine how an organism could develop complicated mouth parts and sensory appendages if there was nothing there for them to actually supply information to.
This was still the engine powering all these functional capabilities, right? Yeah. So you could also argue, just to test out this idea, that evolution stumbled, if you want, into a brain prototype,
and that
suddenly facilitates the computational power to support all these different body machines.
Right.
So you would not necessarily reject that interpretation.
Not at all.
Okay. But now the amazing thing that you also presented to us today is that you actually have found a way, if you want, to come up with sort of more direct evidence that evolution might have progressed in a very specific way, right? That we can go beyond conjecture, but actually look at what brains looked like during the Cambrian explosion, as opposed to having to speculate what they might have looked like.
Yeah, I wouldn't say it progressed in a specific. It's sort of too teleological for my taste. Yeah, I wouldn't know really how to. I mean, clearly there was no progression. There was no internal, engine that drove evolution. These things all occurred by accident and by selection, depending upon their viability. So yes, what very early on they had the evolution gave rise to this thing we call the brain, this simple brain, and that indeed allowed then its elaboration,
right,
through process of hit and miss evolution.
Well, let's just say change, okay? If to keep it sort of neutral in that sense. But I think what was amazing was that you found a way to sort of travel back in time and try and direct evidence for whatever these changes were. And I think this was really, I think, a very dramatic piece of evidence, actually. So maybe
we were lucky. We were lucky.
So maybe you can explain to us what was the question really that you were trying to answer, and how did you answer it?
The question derives from the cladistics that we were doing, trying to reconstruct phylogenies using brain characters, and it indicates that our results in this cladistic analysis indicated that brachiopods could not be the precursors of the insects and crustaceans. There must have been some kind of evolved loss. So again, as I said in the talk, that the only way to prove this is to go back in time and look at the early brains. Now, where did one find early brains? So first of all, I went to the Smithsonian and looked through their fossil collection and found this one particular fossil that showed an early brain. But it wasn't really so satisfying. There were clearly very, very good optic lobes. It had very nice compound eyes.
So how can you see an early brain in a fossil? Because, I mean, the assumption most people have is that brains can't fossilize.
Yes, but that is the assumption. And I never quite understood it, because the brain is the most densely packed tissue in the body. You take an electromyograph through an insect brain or any brain, and it's packed full of profiles. And these profiles have got lots and lots and lots of lipids, and inside there are lots of mitochondria, and there's lots of iron. And it's a very bulky piece of tissue. And if you put that into quite reasonably anoxic conditions, it has a reasonable chance of surviving during mineralization. But there are very few taphonomic conditions that would allow this, and one is in the Burgess Shale, as you know, and the other is in the Xinjiang mudstone. So that's the best way to go to, and you do see these trace internal organs, particularly gut. And gut's very popular amongst paleontologists. They love to say, "Oh, there's gut." But then you look in the head, in front of where the mouth was, and that could be gut diverticular. And indeed, in some specimens, you do have diverticula from the gut that invade the head capsule. But in others, you can clearly see the bundles, the nerves coming in from the antennae, the optic stalk tract going into a structure within the head capsule. And associated with that, you can also see ocelli, you can see the compound eyes, and various other attributes, and also the paired nerve cords. And there you have then evidence of brain.
So this is a fossil of an animal that lived 535 million years ago.
Wapti was 500 about,
right.
Then the ones in the Xinjiang are 535, yeah.
And they're looking quite similar to some living animals.
Yes, the ones from the Xinjiang fauna, the Fuxianhui, its brain is that of a modern malacostracan. Yes, it has the 3 optic neuropils, it has the fused ganglia.
And its morphology, the body morphology,
is incredibly simple. That's the fun part of it. You have this head capsule with eyes that clearly could move conjointly and convergently. They could also rotate, so you had active vision. You have different radii of curvature on the eye, so you had probably acute zones and non-acute zones. So that part of the head, this most frontal part of the head, was reasonably sophisticated, like that of a small shrimp.
Right.
And it has the 3 optic neuropils one would expect from a small shrimp. But shrimps are very complicated. The rest of them have all got these elaborations and spines and interesting appendages and so forth. Whereas Fuxianhui has this homonymous architecture in the thorax and then again in the abdomen.
Mm-hmm.
And one would think, oh gosh, what a simple animal. But no, it's not. It has actually. It must have been quite sophisticated in terms of what it could process up front.
Mm-hmm.
Yeah.
But it did have, let's say, a shrimp-like body, or
it's a crustaceomorph, if you like. It's a stem taxon, which has attributes that one can see to more or less in some modern crustaceans. There's this ankyline species. They're called the remipedes. Godzillogonomamus is Godzilla. I forget how to pronounce this. It's got a wonderful name. This thing is homonymous. Every single segment is identical except the first two segments up front. So it looks like a living fossil, but it's probably also undergone reversal and loss.
Right.
If you look at the brain, it has a malacostricum brain, except it has no optic lobes.
Mm-hmm.
So it has a very superb brain with a very, very simple body. Fuxianhui, which is very ancient, has a lovely brain with a very, very simple body. But that's probably not reduction and loss. That's probably for real. It's an animal that has not reduced anything.
Right.
It's a very early organization.
But now in reconstructing this brain, all you can go for is, let's say, volumetric information, right? What is the size? What are the sizes, relative sizes of things?
No. Volumetry is not really terribly interesting. What's interesting is to actually identify these centers, the three nested optic lobe centers, which are diagnostic of modern malacostricans and insects, but not of brachiopods. So it shows that this organization is the ancestral organization and appeared before the brachiopods even appeared in the fossil record. And also you can actually show from the incoming nerve bundles from the antennae, from the second antennae and from the eyes, that you have then three fused ganglia.
Yes, so how did you identify exactly these three layers?
The three bundles?
Yeah.
The antennae are beautifully preserved, as are sensilla on the antenna. And then out of the antenna you have this beautiful darkly brown stained tract, which is the antennal nerve entering the outline of the brain. And just after the entry point you have this area of rough preservation which corresponds to the olfactory lobe position.
Right.
And that's bilaterally symmetrical. And then a little bit more caudally you have another nerve entering, bilaterally symmetrical, and that nerve it then turns down and plunges down into the substrate, into the matrix of the fossil, and its direction is towards the next pair of appendages, the second antennae.
So the shrimp brain, the modern shrimp brain, is a lot like this very early animal brain. But and you say the shrimp body is much more complicated. Does that mean that a lot of the nervous system complexity that goes with that new body or change to the body is not in the brain, but it's in the other segments?
I think it's probably certainly there are ascending pathways from the ganglia throughout the body that reach the brain in a shrimp and in an insect, and probably in Fuxianhui as well. It may be that some of the more complicated and more elaborate appendages of the shrimp, of the modern shrimp, send information that is required for cerebral computations, tactile information, for example. And that would then involve maybe additional brain regions that have evolved through time, or maybe enlargement of regions that were already there in this ancestral brain. We have no idea. But when we compare across contemporary species, we can see certainly that the brain varies in size and in dimensions of certain regions, not because of actual attributes of the head, but with regard to attributes of the thoracic ganglia and abdominal ganglia.
Right. But I mean, this idea that of a distributed nervous system where within the segments, the local control, for instance, of an actuator system or a limb is happening in that segment. And then the information that's ascending to the brain may be fairly limited compared to what's being sent to the segment. So you're just sending to the brain the information that's relevant to the decisions you might need to make.
Well, yes, the reafferent pathways to the brain certainly will inform the brain about what is going on in the ganglia. And one of the attributes of of a dexterous insect, like say a praying mantis, is the ability to break symmetry. And the circuits in each ganglion, of course, are symmetrical, and they provide symmetrical or at least alternating movements. So to break that is a function of the brain. So the brain is required to, if you like, receive information about the activity of those centers. Absolutely. So the more elaborate, if you like, motor actions that are performed by the limbs, the more reafferents one might expect reaching up to the brain into some of these higher centers. This kind of elaborate dexterous behaviors amongst the crustaceans is quite limited, say, to the crabs, and to some of the malacostricans, for example, the mantis shrimps. And interestingly, the mantis shrimp brain is very, very insect-like. It has centers that one would actually expect in a praying mantis. It's really fascinating. Whereas the shrimp brain, the shrimps, they don't really have much dexterity except for two limbs, and most of the stuff is happening, I think, locally. Most of the motor coordination is happening locally without the participation of the brain.
So I want to pursue a bit more this issue of similarity, right? Because here we have this fossil that's 535 million years old, and it looks like a modern shrimp, if you want.
No.
The brain.
The brain, yes.
Okay.
Yes.
You already explained that the body is not
very
identical,
different,
right? But still, in terms of now, how specific can we get about the similarities of these brains? Like, for instance, if you would look at, okay, now we have the antenna providing inputs to a deposit that matches something like an antenna lobe. Would you say that, let's say, the volume of that antenna lobe and the number of glomeruli that we could imagine would be also housed in that, or making up that antenna lobe, would be comparable to that what you would find in a matching in shrimp's brain?
I think when I would hesitate a very long time before I would try to do that kind of comparison. For no other reason that when you compare the the olfactory lobes across different species of malacostricans, of decapods even, then you have a lot of differences of packing of glomeruli, of sizes of glomeruli within the same volume. So I don't think one can actually try that trick on fossils.
No, so that then puts a boundary on let's say, a similarity assessment you can make.
Absolutely.
Right, and I will try to understand that boundary. So where would you draw that boundary? Just let's say on core structure, the main neuropils making up this nervous system.
I think, as I said, at the moment the boundaries would be to determine the number of segments involved in the brain. If one is very lucky, the position of the olfactory lobes. If one's luckier, then perhaps protocerebral outgrowths, which might suggest a hemiellipsoid body. Certainly the number of optic neuropils, and then the roots of the various nerves that invade the brain, and that's about it.
Right. Okay.
Yeah.
So then, what's the next step in this process? I mean, you're going after more fossils, I assume.
Yes.
Right.
Yeah. So there is the question of the other brain. And the other brain is the brain that pertains to the chelicerates, which are the spiders, the scorpions, and their relatives, including limulus, which is a very ancient morphology. I maybe don't have time to go into all the nitty-gritty of why these brains look so very different from those of crustaceans and insects, but they are very different.
Could you just mention a few outstanding, the most obvious differences?
Well, the most obvious difference is the condensation of both the pre and post oral neuropils. So what has happened is that the first three segments of the brain are pretty much fused with the second, third, and fourth segment of the brain, which is subesophageal, which is post oral, which are pretty much fused with the remaining ganglia, which have come together as one great lump. So at least the brain is a massive neuropil with a hole through for the gut. And it's very different from that of any crustacean or insect. only spiders have evolved a visual system with 3 discrete centers. And these look very different from those of an insect. So there's a very nice example of convergent evolution, although these centers are retinotopic. So there's already major differences. So in the more basal chelicerates, we will never see 3 optic centers.
So the goal then is to now find a fossil that can
match that template, match that search image. But we know from things called sea spiders, Pycnogonidae, their brains have all the neuropils that are characteristic of the spider and chelicerate brains, which have got certain differences from those of crustaceans and insects. For example, the central body complex looks very different in all the crustaceans from those in crustaceans and insects. So the pycnogonid has a spider-like brain, but it has segmental ganglia. So it's the only chelicerate with segmental ganglia that's alive today. Limulus does have segmental ganglia, but they're very, very fused. So we would look then for this spider-like brain plus segmental ganglia in something like Naroe. And as I showed you today, we have one specimen of Naroe which we have very nice segmental ganglia preserved. We haven't got a Naroid which shows the brain yet. So that's the next target. Because the Naroids are supposed to be the stem group arachnomorphs, belong to the stem group arachnomorphs. So that would be fascinating to find the Naroid brain and show that it is actually very different from that of the crustaceomorph brain, the Fuxianhui brain. And then we'd have the other noesis of the other branch of the arthropod.
But what does the Naroid look like? What kind of animal?
It's another very simple animal with a pair of antennae with a head shield and rather simple segments. It was probably much more ambulatory, or rather not so much of a swimmer. We don't really know. But there are certain aspects which distinguish the Naroid from Fuxianhui.
Right.
But there's so much ambiguity of these forms. I mean, look, you saw today Waptia. Waptia is this very strange animal, which, there's a children's book called Animal Crackers. I used to have it when I was a kid. So you had pictures of animals that were cut in 3 pieces, so you could actually mix and match. You could have a catogoose or something like that. So Waptia looks like one of these, kangarillas. We have bits of trilobite and bits of insect and bits of crustaceans all stuck together. So it's very, very hard to say, well, this must belong to this particular trajectory or that particular trajectory.
But now these 2 brains, so one you identified, the second one you're looking for.
We have the search image for
exactly.
that. Yeah.
So you would expect it also to be around 500 million years old, more or less.
535 million. It's got to be the same.
Okay.
It's got to be coeval.
Exactly. But then your suggestion is that these 2 brains are then one bifurcation removed from, let's say, the common ancestor brain.
They have derived from a common ancestor somewhere deeper down.
And that common ancestor, if you go far enough back, was also a common ancestor to us.
Yes.
And so what are the prospects of finding out more about the shared common ancestor between vertebrates and invertebrates?
From the fossil record, I would be very pessimistic. But from modern molecular developmental biology, there is already fabulous information about how you can rescue the forebrain, say, of a Drosophila using a gene that's required for forebrain development in a mouse, and vice versa, homologous genes. So the genetic evidence is that the head segmentation of flies, and therefore, of course, crustaceans, etc., and those of mice, therefore, of cyclostomes, etc., they derive from a common ancestor which has a tripartite brain. That is fascinating, and it's really so exciting. And people like Heinrich Reichert and also Frank Hirth, who have been involved in this work, really deserve a lot of praise for bringing us to that level of understanding, I think. Of course, there are still opponents of this.
But that's an important point, right? Because you use this notion of tripartite brain, which was also used by people like MacLean to say, like, the brain evolved in 3 stages. But it's important to emphasize in this case that the 3 stages are there from the beginning. And not
maybe it did evolve in 3 stages before the origin of these various trajectories. It's possible. we have,
Right.
Be a very different decomposition than one McLean imagined.
No, but since it's the same keyword, I just wanted to sort of emphasize a little bit that we're talking about a different kind of tripartite scheme.
He was talking about amphibians, reptiles, and mammals.
Exactly.
Yeah,
So, and then you just sort of glued one module on top of the other.
That's I think.
So it's not very.
Whereas what we now know is that the history of this structure is very, very far back. In the Cambrian and possibly in the Precambrian times.
Right, exactly.
There's been a very lovely paper from Sten Grilner's lab early this year, demonstrating that in the cyclostome, the organization of the basal ganglia is really no different from that in us.
Right.
I mean, these ground pattern organizations are the indicators of genealogical correspondence, and that's what one's looking for to find, what did the common ancestor really look like in terms of its neural organization.
So Nick, this was a beautiful tour through the, let's say, evolution of the brain, trying to understand its origins. And so to conclude, there are two questions. Also, given your experience, trying to understand really the basic design of brains, what's the Nick Strausfeld law that we should adhere to?
A Strausfeld what?
Law.
Oh, I don't make laws.
You're against law.
Absolutely. Yes. Yes. Ideas, but not laws.
It's an heuristics then, suggestion.
Well, as I said, the next undertaking will be. I wear several hats, as you probably realize. The next undertaking, with regard to the search of will be the other brain, the arachnid type brain. That's one of the undertakings. But then my bread and butter research is on vision, so that's
something else.
That's something else, yeah.
So now, in 5 years from now, we're going to go visit you wherever you are on this planet, digging for fossils somewhere, and we're going to remind you of a hypothesis you're going to generate today, which is like, hey, Nick, you predicted September 2012 that X would be valid in our understanding of the brain. So what's that hypothesis you feel most strongly about today?
Well, I think, as I said, there are the two evolutionary trajectories giving rise to these two major groups of arthropods, the chelicerates and the others, the mandibulates. All these wonderful body forms one sees throughout the history of evolution. They're like decorations on two kinds of Christmas tree. You have the same kind of Christmas tree you have one species like a spruce, beautiful branch Christmas tree for the trajectory leading to insects and crustaceans, another kind of tree towards the chelicerates. Then you hang different kinds of baubles every Christmas on these trees, right? And each Christmas you have a different species, but the basic structure inside is the same throughout. It's a continuum.
And we hang again off another tree.
Right.
Okay, Nick Strausfeld, thank you very much for this conversation.
You're very welcome.
The CSN podcast was produced by the Convergent Science Network of Biomimetics and Biohybrid Systems, a project funded by the European 7th Research Framework Programme. For more interviews, recorded lectures, or upcoming conferences in the field of biomimetics and biohybrid systems, go to csnnetwork.eu. And thank you for listening.